structure of cells and tissues. A lot of people probably are kind of wondering what exactly is oral histology and embryology. So basically what we're going to do is I'm going to take you guys through the very beginning of our development as an embryo, and we're going to start these first few chapters. Like I said, chapters 1 through 4 are a little bit heavier material. So we'll go through those chapters, and things will probably get a little bit more dentally related, because then we're going to talk about the development of teeth, and we're going to then go into the teeth and the oral mucosa and the oral cavity, and all of that stuff is the back part. So, you know, kind of bow up for these first 4 chapters and know that these are going to probably be the more challenging information. And then hopefully once we get this base knowledge, then things will become a little bit easier for you. So we'll start with development and structure of cells and tissues. Now, some of you have probably had biology, physiology, anatomy, those kinds of things. So hopefully some of this will be material that you're familiar with and it's not completely foreign to you. So cells, the human body is composed of cells, and in between those cells, there's intracellular substances that kind of hold all those cells together, and fluid. The cell is the smallest living unit capable of independent existence. And you'll see listed here, these are basically the functions of the cell. To absorb, assimilate, respirate. They have the ability to be irritable, conduct growth, reproduction, and excretion. Cells are composed of a nucleus, which contains the nucleolus, cytoplasm, and in that cytoplasm there are organelles and inclusions, the cell membrane that kind of holds the whole thing together. The intracellular material that surrounds each cell is found in all the cells, and it provides nutrition to the cell, it takes up waste products, and it also provides form for the cell. And here is a little image of the cell, which you guys probably are very familiar with. And you can see all of the little arrows are pointing at all of the organelles that are housed within the cell in that intracellular fluid and material. We are going to talk about each one of those organelles. The cell nucleus is found in all cells except for red blood cells, mature red blood cells, and platelets. There's usually only one nucleus per cell, and in that nucleus is where all of our DNA or chromosomes are located, our genetic material. The cell cytoplasm contains structures that are needed for absorption and creation of cell products. Cytosol contains the organelles and solutes. So this is all the cytoplasm area here, and you can see all the organelles floating around in it. The endoplasmic reticulum is one of those organelles, and it is a parallel membrane-bound cavity in the cytoplasm. And you can see this little kind of layered area off to the right of the nucleus. That is the endoplasmic reticulum. There are 2 types. There is smooth, which is on the top. You can see it doesn't have the little dots on it. And there's rough. And rough is rough because it's got these little dots, and each one of those dots is a ribosome. Ribosomes are very important in protein production. So the endoplasmic reticulum is either smooth or rough. The rough is covered with those ribosomes. Those ribosomes will translate those protein or genetic codes for proteins, and it activates the mechanisms for protein production. So I always tell my students, if you see ribosome, it's important in protein production or something. So it's always going to be associated with that. These may be found as separate particles in the cytoplasm on their own. They can be clustered, or again, they can be found on the surface of that endoplasmic reticulum. The Golgi apparatus is sometimes called the Golgi complex, and the endoplasmic reticulum, that little layered guy down here, is connected to that Golgi apparatus by little small vesicles. It is composed of cisternae, or flat plates, or saccules, small vesicles, and large vacuoles. And the function of the Golgi apparatus is it sorts, condenses, and packages. So I think of that as like the UPS guy of the cell. So it's going to sort, condense, package, and deliver proteins from the endoplasmic reticulum to the cell membrane for export from the cell. Lysosomes I think of as the garbage men of the cell. They contain acid hydrolase and digestive enzymes that will break down substances not only on the inside but outside of the cell. It'll get rid of bacteria and parts of injured cell or an entire cell if it needs to. It is found in all cells except for red blood cells, and it is prominent in macrophages and leukocytes. The mitochondria, you may have always remembered, people usually remember him as the powerhouse of the cell because that is where ATP is generated, and it's generated through the Krebs cycle. The microtubules that are found, that you'll see here in this image, are small tubular structures within that cytoplasm. They are composed of protein, tubulin, and their function is basically just the structure and shape of the cell, and it also has some force-generating elements. The centrioles are the short cylinders that appear close to the nucleus. They're microtubule-generating centers that self-replicate before mitosis begins. The plasma membrane is also sometimes referred to as the plasmalemma, and that is just that external boundary of a cell. It is composed of lipid and protein elements with small amounts of carbohydrates, and that is where signals from hormones and neurotransmitters are received. We're going to talk about this little cell cycle, this little pie that we're looking at here. So the cell cycle is a continuous series of steps by which a cell divides. The first type of cell is a continually renewing cell that line the GI tract and compose the epidermis and bone marrow. The second type of cell is part of an expanding population. So these are cells of the kidney, liver, and some of our glands. And then there's a third type of cell that does not undergo cell division or DNA synthesis. But when we talk about the cell cycle, we're not just talking about mitosis. Mitosis is just a piece of that pie. The cell cycle is the G1, S, and G2 phase, and then that little piece of the pie is mitosis. The G1, S, and G2 phase are all referred to as interphase. And in G1, there's rapid growth and metabolic activity, and centrioles are replicating. And in the S phase, it is important to know that that is when DNA is being produced during that S phase. And then in the G2 phase, growth and final prep for division is happening. So it's kind of resting, getting ready for mitosis. Mitosis is divided into 4 phases. You may have learned it as PMAT, prophase, metaphase, anaphase, and telophase. So all of those little phases are when a cell is dividing. So that is happening during that mitosis cycle. Mitosis happens with our somatic cells. So these are all of the cells of your body with the exception of our sex cells. And mitosis ensures that cells contain all of the same genetic material as the original cell. So it splits off and creates another one just like itself. During meiosis, this is going to ensure that the cell contains half of the genetic material that the original mother or father cell contained, because those cells will then come together, a sperm and an egg, and together we'll have 23 chromosomes from one of the parents and 23 chromosomes from the other parent, and that will give us the full 46 chromosomes. So like I said, mitosis, in that cell cycle, when we're talking about mitosis, it's just that little piece of the pie. The first phase of mitosis is prophase, and during prophase, the chromosomes appear condensed, and the nuclear envelope is not apparent. So things are kind of starting to gather. They start to line up. Each chromosome will split to form 2 chromatids and line up at that equatorial plate. And the pairs will duplicate along with the chromatids and migrate to opposite ends of the cell. So it's starting to divide up because it's going to create 2 cells just like itself. Then metaphase is the second phase of mitosis. Chromatids, as you can see in this picture, have moved to the center. 2 chromatids comprising a chromosome will become attached at that equatorial plate to a centromere. The centromere is kind of the waist of those 2 chromosomes where they gather together. And then the chromatids will split at the centromere into 2 sets of chromosomes. And you can see that in this image. The anaphase stage. This is where daughter chromosomes move to the opposite ends of the poles with 46 chromosomes at each end. Thought to occur by movement of the tubules, attracting those chromatids to the poles, the opposite poles. And then you'll start to see constriction appear around the middle of the cell. So right where those chromosomes are splitting down the middle, you'll start to see it kind of cleave in. And we'll see more of that here at telophase.
I have a question.
What
is a daughter chromosome?
A daughter chromosome, I'm trying to find where that was.
It's in anaphase.
Thank you. So daughter chromosomes, basically when they're talking about that, is they're just giving a name to those separate chromosomes when they split. I don't know if that helps.
Okay, so it's just another name for like how they're separated?
Right, because they were together and now they're splitting apart, and that's what they're calling the chromosome now that is separated.
Okay.
I view it like the product of anaphase. Like whenever it splits, it produces like a daughter cell.
Right.
Oh, okay. Thank you.
Okay, so telophase is the chromosomes now have detached from those tubules that were holding on to them. Those microtubules just kind of disintegrate. The chromosomes will elongate and disperse and regain their chromatin thread of their own identity. And as those little nuclei mature, you see the cleavage furrow deepens in the middle of those 2 cells, and the 2 daughter cells will separate. So now we're calling these daughter cells because these 2 cells are separating and we're forming 2 daughter cells from the 1 original cell. In meiosis, this is for division of sex cells or germ cells, you may hear them called. So during meiosis, there's a reduction in the number of chromosomes to half the normal number that we see with our germ cells. So 23 chromosomes from 46. So our somatic cells had 46. Now our sex cells only have 23 because we only need 23 from mom and 23 from dad. There's 2 cell divisions that happen in meiosis. The first division, the chromosomes will divide equally with pairing of homologous chromosomes and the synthesis of DNA. In the second division, DNA is not synthesized. There are 3 daughter cells that divide into polar bodies that become inactive. and result in only one remaining germ cell that contains half the DNA pairs with the germ cell of the opposite sex. So only one of the germ cells becomes active, the other become inactive. And here is a flow chart that I think is kind of helpful for that. We start up here with the original sex cell with half the chromosomes from mom, half from dad. We go through first meiosis. There's DNA replication and recombination at this point. If you look at the next division, you can see how the blue chromosome pair has a little red leg and the red chromosome pair has a little blue leg. Then they'll split into 2 cells and divide again in second meiosis. At this point, DNA is not replicated. Now those chromosomes split and they are separated into sister chromatids now. And now they will go through that cleavage like we saw in telophase and split into 4 daughter cells. Become inactive, 1 will be active. So that is how meiosis kind of breaks down. This chart I think is more helpful to kind of visually see how that happens. Apoptosis is programmed cell death. So a normal cell will die over time. So fragmentation of a cell into the membrane-bound particles, they are eliminated through that phagocytosis by specialized cells. That cell death occurs during embryonic growth and differentiation when those cells are growing, and then when they differentiate, that's when they're kind of specializing into what kind of cell am I going to be. You're going to be a skin cell, you're going to be a liver cell, you're going to be a nerve cell, and cells are constantly replicating and reproducing and changing. So this eliminates transient and obsolete structures. So our body's always kind of recycling those. And apoptosis is that programmed cell death that happens with those cells that regenerate themselves. There are some periods of prenatal development. The proliferative period is from 0 to 2 weeks. So here we are, now we're talking, we've gone through meiosis, we've got a fertilized cell, or a fertilized egg. So we have a cell, a blastocyst is what we're going to call it at this point. So this proliferative period is 0 to 2 weeks. Implantation occurs and enlargement of this blastocyst, which looks very much like this. You can see it's just like replicating lots of cells and growing, growing, growing. It's implanting, and it implants into that endometrium, or the wall of the uterus. At this point, it contains embryonic tissue. This occurs very rapidly, and this period is only 2 weeks in duration. So in this time, we've got the fertilization of the egg, we've got implantation of this blastocyst, and formation of an embryonic disc will all take place during this period of prenatal development. And here is an image of kind of where this is occurring. It occurs in the distal end of the fallopian tube. You can see the ovary, which is where the egg comes out, and it's fertilized down farther in, a little bit down into the fallopian tube, but it's on the distal end. So sperm cells meet up with the egg, and then you can see that that 1 cell now becomes 2 cells, becomes 4 cells, 8 cells, and so on. When it gets down into the uterus is when it's starting to implant into the wall of the uterus, and that's when we call it a blastocyst, and then an embryo, as you can see by these images. Then we have the embryonic period. So after week 2, that mass of cells, the blastocyst, will now take form as an embryo. That's when we're going to call it that. Different tissues develop, organize, and form our organ systems at this point. So we're talking week 4. We've already got a heart that's beating. And then from weeks 4 to week 7, we've got face and oral structures developing. And it is during this embryonic period, and this is important to know, that tissues are most susceptible to defective development during this time. So the weeks 2 to 6 during that embryonic, or 2 to 8, I'm sorry, during that embryonic period is the time where tissues are really susceptible to defective development. Then we have the fetal period, which is from that 8-week time to 9 months. So that's the rest of the pregnancy. So the embryo has a more human appearance at that 8th week. Tissues are enlarging, they're specializing or differentiating, and they're becoming capable of function. The ovarian cycle, fertilization, implantation, and development of the disc is kind of what we're talking about here. So the origin of tissues, like I said, begins with the fertilization of the egg in that distal portion of the uterine tube. The fertilized egg at that point is termed a zygote. The production of a morula, or that ball of cells, I think of this ball like snowballing as it's rolling through the fallopian tube. The cells are multiplying. And then when it comes into contact with the uterus and implants, it's a blastocyst. And then I believe it's week 2 that we're now going to call it an embryo. So we've got this morula, and that occurs in the uterine tube and migrates medially towards that uterus. The uterine cavity prepares for that fertilized ovum by thickening, and that's the endometrium that thickens. And then the capillaries and glands will develop to nourish the ovum as it attaches to that endometrium.
So a morula is just a ball of cells?
Right, like when it's up in that distal end and it's very first fertilized and you see it, it's 1 cell, it's 2 cells, it's 3 cells. They're calling that a morula until it reaches the uterine wall, and then it's a blastocyst and attaches to the wall. And then after 2 weeks, they call it an embryo because it starts to take on, it starts to differentiate.
Okay.
So as the morula enlarges, it's a blastocyst. The blastocyst attaches to the sticky wall of the uterus, and its cells digest the uterine endometrium, permitting deeper penetration, and that is called implantation. Many women will actually experience bleeding at this point, and I can tell you that that did not happen with my son, but it did with my daughter, and I almost wonder if it wasn't hormonal because she was a girl. But it's a scary thing for a lot of women because you really think that you're losing the pregnancy, but it's fairly common thing, so that can occur. Okay, so 2 small cavities will develop on either side of the inner cell mass, and this is like an embryonic disc. It'll form between these 2 cavities, and that becomes the embryo. And here's an image of that. So if you can kind of, it looks like 2 kidney beans to me. One side becomes the ectoderm. That middle section is what's known as the mesoderm, and then the endoderm. Ecto, when I think of ecto, I think of outside. So it is the epidermis, so the skin, the hair, the nails, tooth enamel. Those kinds of things all develop from the ectoderm. There are a couple of other things that develop from it. The nervous system, sensory epithelium of the eye, ear, nose, mammary and cutaneous glands, the epithelium of the sinus and oral and nasal cavities. But I think of the ectoderm more as outward things. The mesoderm, the muscles, connective tissue derivatives such as bone, cartilage, blood, the dentin and pulp and cementum and periodontal ligament of the tooth is important to know that it comes from mesoderm. And then the endoderm is easy because it's one thing. It's basically the inside of it's your GI tract from your mouth out, and all of the glands that are associated with that. So this is this little embryonic disc at this point that is forming from that little blastocyst now that has attached. Epithelial tissue is skin, basically. We've got the epidermis and the dermis. Epidermis is that surface cell layer that develops from those ectodermal cells, and the dermis develops from the underlying mesoderm. And there's a great picture in your book that you can look at here and kind of see how that's all divided and what those layers are. Initially, the embryo is covered by only a single layer of those ectodermal cells. In about the 11th to the 12th week, this layer thickens into 4 layers, and the superficial layers of epithelium come from basal layer of cells. So superficial is towards the top. Skin, when it's reproducing, is reproducing from that basal layer or the stratum basal up. So that's why, you know, they talk about, if you've ever seen those commercials that talk about how much dead skin is in a mattress after so many years, that's kind of disgusting. But our skin reproduces from the basal layer and it pushes out the old cells. So that top layer sloughs off every so often, and quite often actually. And so it's constantly making new cells and creating new skin from that basal layer up. Later, after, you know, the cells are being produced and we're making new skin, melanocytes will invade that and give pigment to the skin. So that's what gives our skin the color it is. The nervous system is the brain and spinal cord, and there are neural folds that begin to appear during the 3rd prenatal week. These folds will arise dorsally along the lateral edges of that neural plate, and the neural tube will close anteriorly and posteriorly, and that becomes our spinal cord, basically. That tube that we see kind of running down this little bean. Connective tissue develops from somites as fibroblasts that migrate from either side of that neural tube. There's the medial sclerotome, which differentiates into mesenchymal cells. The mesenchymal cells will become osteoblasts, which build bone, chondroblasts, which build connective tissue. I'm confusing myself. Fibroblasts are connective tissue. Chondroblasts are cartilage, I believe. But the medial sclerotome differentiates into these cells that create the osteoblasts, chondroblasts, and fibroblasts. The dermatome form the dermis, subcutaneous tissue, the visceral mesoderm, and support the endoderm of that GI tract and the system of mesenteries that will stabilize and support that GI tract. Blood. Blood is a specialized connective tissue. 7 liters of fluid and cells make up blood in a human body. The red blood cells are also called erythrocytes, and those are the most numerous that we have in our body. They are in charge of carrying oxygen from the lungs through our hemoglobin. They carry carbon dioxide from cells of the body by hemoglobin of the red blood cells and plasma of the blood. And then our white blood cells are called leukocytes, and they're important in inflammation and immunity, and we're going to learn a lot more about that later, especially in pathology. Blood platelets are thrombocytes, and they are in charge of blood clotting. And then plasma is just the liquid that the cells float around in our blood. White blood cells function in the defense of the body. Like I said, they're the first responders to infection. They will migrate between cells and tissue spaces to the site of infection. We have the lymphatic system, which is really important for infection as well. That's how infection drains in our body. It's composed of the lymph nodes, the thymus, and the spleen, and then all of the vessels that carry that lymph fluid through our body. You can see that lymph is all over our body. So many times you'll hear people talk about lymph nodes and lymph involvement with cancer. If cancer is infects lymph nodes, you can see how that can travel all over your body really, really quickly. So that's in staging and grading cancer, that's one of the things they look at is how many lymph nodes are involved. Immunologic defense of the body is important. Like I said, with lymph, it destroys bacteria, viruses, and any invasive microorganisms. So it's a really important system in our body for protection against infection. Cartilage and bone. The initial skeletal component in an embryo is cartilage. So that's why babies can, you know, take their legs and wrap them up around their head, and they're very flexible because their bone is not hardened yet like it does as you get a little bit older. So these cartilage cells arise from the sclerotome and migrate to surround that notochord and spinal cord, which again is the spinal column. The skeleton forms in segmental pattern, and our muscles do the same thing. So if we look at this picture on picture A here, where it shows calcifying cartilage, you can see how it's a very primitive little bone and it's soft. It's not calcified yet. So in time, we start to have the blood vessels will invade that bone, and then it'll start to be calcified and replaced. That cartilage will be replaced with bone as that baby grows. And then if we look at D, you can see how it has the epiphysis, or the ball on the end of the long bone. The metaphysis is in that ball and the long shaft, or the shaft is also called the diaphysis of the bone. So the epiphyseal plate will occur right there where the metaphysis is. And that is when they talk about, you know, growth plates in teenagers, that area between the epiphysis and diaphysis will remain open. So if you were to take an x-ray of that, it'll look radiolucent until that patient has reached their full height, you know, mainly in your long bones. So like your femur or your tibia and your fibula. So they can tell if a child is still growing, or if a teenager is still growing, by whether those growth plates are closed or not. And many times injuries will happen at this area because it isn't closed. So that's kind of what we're talking about with bone when you hear people make those comments. Appositional growth is exogenous. It occurs on the surface. Exo means outside, ecto means outside. So appositional growth is occurring exogenously or on the surface of the bone. So new bone would form along that cartilage margin of the epiphyseal line. The bone would replace the epiphysis. Cartilage is then limited to covering the heads of the long bones, the nasal septum, and our ears. Interstitial growth is endogenous, so that's going to appear inside the bone, and what is happening is a proliferation and expansion of those cells within the bone, within that bony matrix. Supportive cartilage skeleton is produced rapidly, and it will support the soft tissues of that growing embryo. Most of cartilage skeleton is later replaced by bone. So all that cartilage that we see in the little babies, most of that over time is going to be replaced with bone, because bone is going to give their body frame and structure and strength. So that's what happens as, you know, as the baby grows. Okay, so intramembranous or membranous bone formation. This is a direct transformation of connective tissue into bone, where collagen fibers of that connective tissue will organize and form like a meshwork, which gradually calcifies. We see this in the bones of the cranium and face. They form through membranous or intramembranous bone formation. Muscle. During the 10th prenatal week, we see muscle cells or myoblasts. Anytime you see the prefix myo, they're talking about muscle. This is where if we had med terminology is helpful because you can use prefixes and suffixes from words and often figure out what that word means. So muscle cells will migrate from the myotome in a segmental pattern. They'll differentiate into elongated multinucleated fibers. Each entire muscle is covered with what's called paramecium. And then inside of that, you've got little groups of muscle fibers called muscle fascicles, and those are covered with epimecium. And then each individual fiber is covered with endomecium. There are 3 types of muscle fibers that we talk about. Skeletal muscle, and this is controlled by our voluntary, they're under voluntary control, and they are striated. Smooth muscle is involuntary. So smooth muscle would be any muscle of our internal organs and things like that. Skeletal muscle, obviously, are the muscles that we use to move our bones, our leg. We have control over that. Anything you have control over is skeletal muscle, and it's striated. Smooth muscle is involuntary, and then cardiac muscle is the kind of an exception. It is involuntary as well. And you can see that it is also striated, but it's involuntary. We can't control it. Skeletal muscle will lose that segmental pattern as it acquires insertion or skeletal elements. It becomes striated, voluntary at that point, and it will divide and supply dorsal and ventral parts of the limb. So you'll see it connected to the dorsal and ventral parts of our limbs. It provides deep superficial muscle fibers. And that striation that we talk about are just those lines that cross that muscle to give it those sites of contraction. Cardiac muscle will pump blood through 50,000 miles of blood vessels. It has long cross-striated fibers. Like I said, it's striated but involuntary, and it contains what we call intercalated discs that help with its contraction. This is found in the wall of the heart as well as the major veins that open into the heart. And then, like I said, smooth muscle is what controls mainly our GI tract. It supports our trachea, our bronchi, our urogenital tract, and our vessels. Muscle cells will develop and become oriented in the direction to which their contraction will be exerted, and this is under the control of our autonomic nervous system, which is involuntary. Cardiovascular system will originate from angioblast. Angio means heart, another prefix. The angioblast will separate into clusters. So we've got the outer cells that will organize into a series of tubes, and then the inner cells will become blood vessels. And after those first few weeks of nutrition, the first few weeks of nutrition are provided to that yolk sac, through the yolk sac to the embryo through what's called the vitelline vascular system. And then at about 4 weeks, those vessels develop and form a vascular network that takes over for that vitelline vascular system, and you'll know that as the umbilical system, the umbilical cord. So that will then connect after 4 weeks to the placenta, and that will provide nutrition to the embryo at that point. Developmental defects can occur, as we know, and those can either be due to environmental reasons or they can be hereditary. So a developing human is least susceptible to teratogens during the proliferative period. So remember that period where we've got the morula, the blastocyst, we're not an embryo yet. That is the time period that is least susceptible to teratogens. The embryonic period is that 3rd week to the 8th week, or I think the book might have even also said 2nd to 8th week. But this is the most critical time in development because this is where the cells are really differentiating, specializing, things are starting to actually function. So this is the most critical time for us to be careful. So if, let's say we had a patient that needed to have dental work done, this is going to be the time we're going to avoid. We're going to wait until after that 8th week, or at least into the 3rd or 4th month of pregnancy, if possible, to, use any type of anesthesia or do any dental work or anything like that, unless absolutely necessary. Hereditary causes that could cause developmental abnormalities could be genetic. These would be present from one generation to the next. Chromosomal abnormalities can happen where there's an increase or decrease from the normal number of chromosomes present. Abnormal development can also be caused by an expression of a defective gene. So it could be a dominant or recessive gene. A dominant gene would express itself whether on one member of a pair of homologous chromosomes or both. On a recessive gene, you would have to have 2 sets of chromosomes containing that gene in order for that to be present. So those are some things that can happen and cause hereditary causes for those developmental abnormalities. So which dental environmental factors might have a different role during the first trimester than the last trimester? The answers are right there in front of you. X-rays and anesthetic. And we talked about X-rays with pregnant women in radiography. We talked about how we're only going to do X-rays if the benefit outweighs the risk. So if you have a patient that is pregnant and has, a hot infection going on, we're taking X-rays because that infection could cause more problem and damage for that child that's developing inside of her than that one X-ray if we cover and protect her. Anesthetic is the same thing. We'd like to wait, like I said, past that 8-week period. We want to get past that proliferative period and embryonic period and wait until the 3rd trimester. Okay. How are we doing?
Good. It's just a lot of information.
Yeah, it is. It is a lot. I will definitely tell you that. And like I said, these first 4 chapters, if we can get through these and get our brain wrapped around this stuff, the next, there's still information, but it's going to be more dentally. And you'll probably hear that information not only in my class, but in, I know that Franklin's class and this class in some cases parallel one another. So you'll be hearing some of that information again. Okay, I'm going to go ahead and here's what we could do. I can go on to chapter 2, or we can go through the syllabus and do 2 and 3 this afternoon later. Any requests?
Do you have time to get through all of chapter 2?
Probably.
Okay, that works for me.
You want to do that? All right. Chapter 2 is going to be kind of a continuation of what we've talked about. So now we're going to talk about structure and function of cells, tissues, and organs. So I think of it as we start with cells. Those cells form tissues. Those tissues form organs. Those organs become organ systems. So that's kind of where we're going with this. So we're going to talk about how those tissues of the body will build on one another. And we'll talk about all the different organ systems, and I'll let you read through those. Those are all the different organ systems in our body. Integumentary, respiratory, digestive, you see them all there. Cells and tissues. We'll talk about tissues first. We've got epithelial tissue, neural tissue, which is nerve. Epithelial obviously is your skin. Connective tissues are ligaments, tendons, everything that kind of holds our body together. And then muscle tissue is our muscles. So we'll first talk about epithelial tissue. It is composed of different layers. There's a superficial layer, and hopefully you guys have learned what those meanings are. Deep is, obviously deep down in, and then superficial is what we see on the surface. So our superficial layer are closely packed, covering the external surface of our body. The dermis is that connective tissue layer that underlies the epithelial tissue. So we've got our epidermis that lays on top of the dermis, and the dermis is underneath and kind of connects our skin to everything underneath of it. There's a much thinner layer that lines the internal cavities of the body and tubes that drain glands and carry blood throughout the body underneath all of that. So dermis is closely associated to that epidermis on the surface, and it has interdigitating relationship in some areas where there's little finger-like projections that hold the epidermis to the dermis. There are nerves that exit the dermis, and some will penetrate epithelial cells to function as receptors. And you'll see that in that drawing that I showed you, where it was like there's like a slice of epithelial tissue, and you can kind of see how that is layered. Then there's sweat glands, hair follicles, sebaceous gland, the erector pili muscle. I don't know if any of you know this, but when you get goosebumps, it's the erecti pili muscles that are kind of standing up. They're located in the dermis and the subcutaneous tissue. Here's the picture I was trying to find for you guys. And that, I believe, is in your book, but I'm having trouble locating it right at this moment. But anyway, that kind of shows you like the hair shaft, the sweat glands, the epidermis, dermis, the subcutaneous layer, and how those vessels and everything kind of work in between that. The stratum corneum, stratum germinatum, spinosum, and basal are the layers of skin that you're going to see associated with that. Obviously, basal is easy to remember because I told you skin reproduces from that basal layer. So always remember that that is the deepest layer of that tissue of the dermis. Epithelial tissue is described according to cell shape. So there's different shapes. There's squamous, cuboidal, columnar that you may see associated with it. And then it's also classified according to its layers. If it's simple, it's a single layer. If it's pseudo-stratified, all cells in contact with that basal lamina, but they aren't in contact with the surface. And then stratified, there are several layers. Only the basal layer is in contact with the basal lamina. Here is a picture that I think kind of helps. It shows you, simple is a single layer, the different shapes, the different, what stratified looks like. So I feel like I like to have visuals along with some of this stuff if I possibly can. Epithelial membranes function in one or more of the absorptive processes, and the processes are listed there for you. Neural tissue arises from neuroepithelial cells. There are areas for reception and correlation of messages from our nerves. So we've got neurons, which you guys are learning about in anesthesia. The nerve cells that receive and conduct impulses and regulate the muscle and gland activity. So our glands are controlled by our nerves. There are neuroglial cells, which are supportive cells of that nervous system. The nerve cell body is also called a perikaryon, and it contains the nucleus and the cytoplasm of the nerve cell. There are sensory ganglia that are just like little swellings that are associated with each spinal nerve. So where those nerves go into the spine, there's like these little ganglia, which are swellings. They're called dorsal root ganglia, and they are associated with our cranial nerves. So they're associated with the trigeminal, the facial, the glossopharyngeal, and the hypoglossal. In that nerve, there's the axon, which is that long, thin process, and it can vary in length depending on where the nerve is located. And the axon carries the nerve impulses away from the cell body. And I always remember that because axon is an A and away is an A. So the axon carries the nerve pulse away from that cell body. And it terminates into axon terminals, or those little synaptic ends. The axons outside the central nervous system are protected by that myelin sheath. And I think you guys were learning about that yesterday. So the nerve axons that are not within the central nervous system, so in other words, not the brain or the spinal cord, all the nerves outside of that are covered with that protective myelin sheath. The dendrites, which there are usually multiple dendrites present, those receive nerve impulses and conduct impulses towards the cell body. And then, like I said, those neuroglial cells protect and support those nerve cells. They are 5 to 50 times more numerous than neurons. Some of these cells are phagocytic and ingest bacteria as well. Here's an image of that. So you can see the axon terminal on one end, and then the long axon with the myelin sheath with those little nodes of Ranvier where they can jump. I think of it almost like jumping rocks from one node to the next. And then the dendrites where messages are received on the other end. Connective tissue, there's connective tissue proper classifications. There's 3 kinds: loose connective tissue, dense connective tissue, and then we have loose connective tissue that has special properties. The functions of connective tissue are tissue support, and they kind of hold things together, protection of our body parts. It's an area for fluid exchange, and also an area of adipose tissue storage. Again, an image showing you where you would find connective tissue in the body. Cartilage, like I said, chondro is the prefix for cartilage. There's hyaline cartilage, elastic cartilage, and fibrous cartilage. Hyaline is the most common type in our body. We see it the most. It's present in our nose, our trachea, the little rings around the trachea, the larynx, the ventral ends, the edges, the ends of our ribs, and then the articulating surfaces of long bones are covered with cartilage for protection. Elastic fibers are the epiglottis, the cuneiform cartilage of the larynx, and then the auditory canal and tube are elastic. And then fibrous cartilage is found in our vertebrae, those discs that are in between our vertebrae. And then they're at some insertion points of our tendons. And then where the pubic bone comes together, right in the middle, that's called the symphysis pubis. And that is also fibrous. And again, kind of telling you again where those are located. So this is a kind of a good chart to look at if you're studying and you kind of want to know and have some visual of where those different types of cartilage are found. In our body, we have a couple different kinds of bone. We have compact or dense cortical bone, which is found on the surface of the bone. So that's the smooth outer surface of the bone that we think of when we think of bone. Cancellous bone is found in the center of our long bone. So if you took a long bone and you cut it in half and you looked at the inside, it looks like a sponge. There's all kinds of holes or trabeculae that run through there. And those holes and trabeculae are areas that nerves, blood supply, and interstitial fluids and things can flow. So that's the difference between compact bone and cancellous bone. Blood, like we said in the last chapter, provides oxygen to our cells, returns carbon dioxide from cells to the lungs. It helps us clot so that we don't lose too much blood. It helps to regulate the pH of our body through its buffering system. It helps regulate our body temperature. And blood also provides protection from bacteria because there's phagocytic capabilities and antigenic properties of our blood. Lymphocytes, again, responsible for our immune system. They circulate in the vessels and bloodstream and help get rid of infection, inflammation, bacteria, all those kinds of things. T cells are prominent in our immune system. And then we have B memory cells and plasma cells. The T cells have surface molecules that call them to action. The B memory cells are the ones that, like right now, we're wanting to know, are our bodies producing antibodies against this crazy coronavirus? The B cells are part of the humoral immune system, and they are the ones that do produce antibodies in response to foreign substances. So they have memory. Our immune system has memory. And that's how we produce these antibodies that will be able to stand up and say, hey, I've seen you before. You're not welcome here. And our body can fight it. Plasma cells produce antibodies in response to foreign substances as well. So that's the plasma therapy that you hear them talking about. I think President Trump actually received some of that plasma therapy. Okay, so muscle tissue is located throughout our body, and there's three types. There's skeletal, like I said, which is helps us move our skeleton, basically is how you can remember that. It's movement under voluntary control. Smooth muscle is involuntary. This is our digestive tract. It's important for motility of the alimentary canal, so it moves things through our body, but we can't control that, so it's involuntary. And then cardiac is involuntary, and that's in charge of pumping the blood through our heart. Muscle tissue features that are common to all muscles are the epimysium, which we talked about, covers the entire muscle. And then we're going to divide that muscle. Okay, now we've got muscle fascicles, which is like a group of little fibers, and that's covered by paramysium. And then the endomysium is on the innermost part of just one muscle fiber. All of the organ systems, the integumentary or skin, that's the largest system. Obviously, if you think about our skin, it covers our entire body, so that is the largest organ system we have. Neural is our nervous system. Skeletal system is our bones. Digestive is all of our GI inner workings. Respiratory is the lung system. Vascular is all of our blood supply, blood flow. Lymph system or lymphatic system we've talked about. Muscular. Endocrine is all of our glands. That is regulated by the neural system. The nerve system has kind of control over our hormones and release of hormones from those glands. The urinary system, reproductive, and then special senses are your eyes, ears, nose, those. Tissues are integrated and then specifically designed to form different functions. So these cells aggregate to form tissues or groups of tissues that are organized together to form organs. And then the organs form specific functions and relate to other organs to form organ systems. Again, the skin system or integumentary is the largest organ in the body, and its function is to excrete waste products, whether that be carbon dioxide, water, salts, or urea. It also eliminates heat through perspiration. It protects from the invasion of foreign materials. That's why we don't like to have cuts on our hands or our skin. And nerves of the skin receive stimuli from outside of the body. Skin is composed of the epidermis and dermis. The epidermis rests on a basement membrane that separates it from the dermis. And then we have the dermis. There's epithelial cells that will form membranes that are composed of closely associated cells and intracellular substance that kind of holds it all together. And there's lots of functions of the skin. There's 5 cell type layers to the epithelium that I was showing you on that picture, and now it kind of shows you on this one a little bit easier. There's not quite as many things pointed out on this image. So if we were going from the deepest layer to the most superficial, that's how these are listed. So we've got the basal layer. They call that also the germinating layer because, again, remember that's where cells, skin cells or epithelial cells are being produced, and then they migrate up to the superficial layer, which is the stratum corneum. So we've got basal layer, then stratum spinosum, and then the granular layer, and then the stratum lucidum, and then stratum corneum. The neural system, or the nervous system, is the central nervous system consisting of the brain and spinal cord. All sensations received in the body are relayed to the brain and spinal cord, which will then act on those sensations. So if the brain receives a stimuli that, hey, that burner's hot, it's going to send out a message to the muscles to pull the hand away from the hot burner. So that's kind of how that works. The spinal cord extends from our brain all the way down to our lumbar vertebrae. There's 31 segments, and in every segment there are spinal nerves that come out of those segments. The spinal cord conveys impulses from peripheral nervous system to the brain. So the spinal cord, all those nerves that go out of the spinal cord receive messages from outside or the periphery, and they send those impulses to the brain. Afferent sensory system and efferent. Afferent carries information from the peripheral nervous system to the central nervous system, and efferent are motor, so they carry information from the central nervous system out to the muscles and glands. So one of my students told me that they remembered efferent as get the EFF out. I know that you can take that in many different ways, but that has always stuck with me and I've never forgotten that. So efferent means out. It is taking the information that it's received and saying, you need to move your hand. So it's sending a message out to the muscles to move the hand. So that's the difference between sensory and motor. Afferent is sensory, meaning it's carrying information that it receives from the peripheral nervous system to the central nervous system, saying that's hot. And then the efferent carries that information and says, you need to move your hand. The somatic nervous system carries impulses to voluntary muscles, so skeletal muscles, which again are under our conscious control. And then autonomic, I think of autopilot or automatic, and this is the smooth muscles and cardiac muscles and glands, things that we don't have control over. Our body just does it on autopilot. Again, autonomic nervous system, involuntary responses. It is then further divided into the sympathetic and parasympathetic. The sympathetic is our fight or flight response. So when the sympathetic division is engaged, your body is going to shut down things it doesn't need, and it's going to kick up the things it does need. So if you're running, it's going to kick up, hey, we need muscles, we need to be able to breathe fast. Our heart's going to beat faster, but right now I don't have time for tears. I don't have time for saliva. I don't need to be worried about digestion. All that stuff's going to shut down because I need to take care of this fight response. So that's sympathetic. Parasympathetic is vegetative function. So this is, I'm just sitting here chilling and my body's taking care of everything else that it needs to do. Like I said, all the digestive, respiratory, motility in the, throughout your body, all of that stuff's happening. That's just on automatic cruise control. Skeletal system is our framework. As you know, all of our muscles and ligaments are attached to our bones. The bone not only helps us move with our arms and legs and all of that, but it also encloses and protects things. It protects our brain, our skull does, our spinal cord, as well as our lung. And it's also an important system because our blood and marrow and things are made in our bone. So it's a hematopoietic system that is also important. Alimentary canal functions. It absorbs, transforms, and extracts any needed components from our food, and then excretes any solid waste. Our pharynx and esophagus conduct food to the stomach. They reduce the food down to what is called chyme. That food then travels to the small intestine where our digestion continues. And most of the nutrients in our body are absorbed in the small intestine. And as you know, we have lots and lots of feet of intestines. It also has secretions from glands such as the liver, pancreas, and spleen that add to our digestive ability and help us break down some of those components of food. And then it goes into the large intestine where very little but some nutrition is absorbed, solid wastes are created, and then released through the anus and rectum. Respiratory function, the main function is the exchange of gases. There's the inflow of oxygen and the outflow of carbon dioxide. The nasal cavities also warm the air and moisten it. The nasal chambers will trap dust particles, and the cilia will move those particles from the respiratory tract to the pharynx. So it helps kind of keep our air clean too. And then the vascular system includes the heart, the large elastic arteries, the smaller muscular arteries, and then all the miles and miles of capillaries and veins. And again, the heart pumps an estimated 5 to 6 liters of blood per minute, which is huge. It's a lot of blood. Lymphatic system, we've already talked about. Lymph nodes, thymus, and spleen. It is characteristic of the immune system. It has memory to those foreign molecules or macromolecules that enter our body so that we can fight them when we see them again. Muscular system, striated muscles, large muscles of the limbs, chest wall, and neck. They enable the actions of our body to stand erect, to walk, as well as any other body movements. So they're made up of cylindrical fibers or muscle fibers under voluntary control. And then there's little myofibrils of that muscle that permit contraction or relaxation of muscles. And this just kind of shows a picture. It's showing all the muscles of the body.
So I have a
Endocrine.
question. So what happens when your muscles like spasm?
When they spasm, I can't explain that to you. I would have to like look that up. I'm
No,
not
that's
sure
fine. I was just curious.
whether it's just like a misfiring where it just, you know, usually it happens due to overuse or fatigue. So I'm not real sure what causes that. I would hate to like tell you something and have it be wrong, but that'd be a good thing to look into.
Okay, thank you.
Oh wait, endocrine system. We've touched on some of these. So the endocrine system is made up of the thyroid, the parathyroid, the pituitary gland, the ovaries, the testes, the pancreas, the adrenal medulla. So all of these glands that I just listed secrete hormones. And those hormones are specific to certain areas of the body, like the thyroid, for instance. It secretes thyroxine, and it is important in our metabolism. So, you know, all of these, parathyroid is important in our body's ability to control calcium levels in our body. The pituitary is growth hormones. So, you know, if you're producing too much growth hormone, you might be super, super tall. If you don't produce enough, you're going to be smaller. So that's kind of what the endocrine system is. Each hormone will act on a specific receptor cell. So each hormone has a job. And some organs of the endocrine system function along with other functions. So the neural system is important in this as well. So they kind of work together in harmony, some of these systems. The urinary system includes kidneys, ureters, bladder, and the whole urinary tract, which filters toxic, unnecessary substance from our bloodstream. And then the kidneys will excrete that waste and bacterial toxins in addition to water. The major function of our urinary system is to control our blood volume and pressure, the composition of urine, and some other things that regulate our blood pressure. But we don't always think about that. But the control of blood volume and pressure is important. The urinary system is important in that. Just a picture of what is included in the urinary system. Reproductive system, male and female. The testes are the ones that secrete testosterone. There's the prostate and seminal vesicles that makes up the male reproductive system. The female are the ovaries that produce estrogen as well as progesterone, the uterus and vagina. Images of those. And then, like I said, special senses. These senses permit us to detect changes in our environment. So vision, hearing, equilibrium, which some of our equilibrium is controlled by the inner bones in our inner ear. Smell and taste also. So why do we need to understand cell structure, tissue, and organ function? Because all of these things play together, hopefully harmoniously, in our body to keep our body functioning well. And we need to understand this because when we have patients that don't have bodies that are functioning well, sometimes the oral cavity is one of the first places that we can detect problems with patients. So it's important for us to understand. That's why we start at the very, very beginning, fertilization. What happens, what can happen along the way that can cause problems dentally or physically that can lead to oral manifestations later. So that's why all of this information is important, but it's a lot of information. And we just have one more chapter, but I'm glad we're dividing it because I feel like we need to maybe spend a little bit more time with it because it's not material that is as familiar to you. So I am going to, let me stop this recording.